Okay, so let us look at this water-soluble ligand cell signaling pathway. So it means that these ligands, they are hydrophilic. Okay, they are hydrophilic. So it means that they are water-soluble. Okay, so but I will go through in general first. So what's been cell signaling or signal transduction? Actually, they got a different. Cell signaling and signal transduction a little bit different. Later, I will show you guys. So it's one of the important process required for the cells when we want to monitor and respond appropriately to their environment. For example, blood glucose concentration. This we're going to learn in A2 again, which involve pancreas, muscle, and liver cell. So you try to imagine, okay, when you have high carbohydrate meal just now, okay, your lunch. So it means the blood glucose level increase. So pancreas detect it. When pancreas that, oh, blood glucose increase already, we have to do something if no diabetes. So pancreas say that, now mind, I want to communicate with the liver cell. I want to communicate with the muscle cell. Then how they communicate? They cannot shout, right? They cannot shout, hello, please do something for to reduce the blood glucose level. They cannot shout. So it means that the communication here, the cell signaling have to take place. So pancreas have to send something to the muscle. So pancreas send insulin, the hormone. You have learned about this, right? Insulin, right? So send this insulin to the muscle cells or liver cells. So this is a form of cell signaling. Are you clear? Okay. So in order to respond to these stimuli in the immediate environment, cell must be able to receive and process the signal. Okay, originates outside their plasma membranes. Are you clear? This signal, okay, because water soluble, right? So water soluble, it must be in the tissue fluids. Okay, so in multicellular organism, for example, our growth factor, the hormone, neurotransmitter. Okay, so neurotransmitter actually involved in neuron. Are some of the many types of the chemical signal, or we call the ligands. Okay, cell will be used. A growth factor is naturally occurring substances capable of stimulating the cellular growth, proliferation. Proliferation basically means that division. Okay, the cell divisions. Okay, healing because of the mitosis and the cellular differentiation. So it means that we want not only they divide, but also they form the particular type of tissues. Okay, so a hormone, another form is a hormone, growth factor, the first. Second, a hormone is a signaling molecule produced by glands in multicellular organism, transported by the blood or cardiovascular system to the target distant organ, regulate the physiology and behavior. For example, insulin, glucagon. Later, we're going to use the glucagon and also adrenaline. Both hormones, you guys are familiar. And we have another class of the ligands known as neurotransmitters involved in the neurons. They are short range in this case, signaling molecule. So what's short range? So it means that we have one neuron, another neuron. So in this case, so it means from one neuron to another neuron in this case. So very short range only. So this one is neurotransmitter. So basically in a cell signaling pathway involving the water-soluble ligands, first we're going to detect the stimulus on the cell surface membrane. Can you see that? And we transfer the signal to the cytoplasm, and with transmissions of the signal to the effector molecules. Are you clear? So we have three what we call these processes. Okay, so let me draw it out to summarize what you need to know. Okay, so now try to imagine, okay, the whole things, yeah. Okay, so I'm going to use one of the example called glucagons, okay, because what we're going to learn later related to glucagon or adrenaline. Okay, so first of all, we talk about glucagon. And you guys are familiar with glucagons, right? So glucagons helps to increase blood glucose level. Means that when we starve for a long time, say the glucagons will help us. Okay, so the target organs here, okay, for glucagon, okay, is liver cell. So liver cell will respond to these glucagons by one of the main process, breakdown or hydrolyze. Hydrolyze what? glycogen. So means that in the process, we want to break down glycogen into many glucose. Cannot hydrolyze, right? So we need to have the enzyme. But this enzyme normally inactive. Are you clear? Not active. But if let's say with the presence of the glucagon, this ligand is a ligand. With the presence of the glucagon, therefore enzyme become activated. Therefore, we're going to release, glucose because of the glycogen hydrolysis take place. Are you clear? Okay. So now, involve this the whole process. You can see that who detect. So detections of low blood glucose. By who? By the pancreas. Or more precisely, by alpha cell in pancreas. Okay, you learn about islets, right? Islets of Langerhans, correct or not? Okay, so try to imagine, so I have this pancreas. Okay, pancreas want to communicate with, okay, with who? With the liver, because liver store glycogen. So here we need to have the communication. So this communication by what? By chemical signaling pathway. Are you clear? And this chemical is glucagon. Now, very important. We want liver only respond. We don't want the whole body respond. So we want to have specific. So glucagons must be specific to liver cell. So how this thing can take place? That's one. Okay, I want it specific to liver cell. Second, glucagon is a water-soluble ligand. Okay, so let us look at the liver cell now. So for this process to take place, glycogen, we want the glycogen to be converted to glucose. We need to have the enzyme. So this enzyme is inactive normally. Can I see that? Are you clear? So this is a liver cell. Okay, so what happened here is, first, we have the issues. First, I want to be specific for liver cells to respond. Second, my target here is this enzyme. But my hormones or ligand is a water-soluble ligand. So water-soluble ligand always have the problem, they cannot or unable to diffuse across cell membranes. And they are, okay, they are relatively larger compared to the small molecule. Can I see that? So what will happen here is to achieve the specificity, as well as to allow the signal transduction here, what will happen? So on the surface of the liver cells, you will have what we call as the receptor. Okay, so this is the receptor. So depend on what kind of the hormone. If this is for the glucagon to bind, if let's say this is a glucagon, so this receptor is termed as glucagon receptor. Can you see that? Are you clear? But the thing here is my effector, my final molecule that execute the events for us is inside the cell. So we need to have the signal transduction. So it means that throughout this process, okay, so this process will trigger a series of reactions, and then we activate the enzyme. Are you clear? A series of reactions until we will be able to activate the enzyme. So when activate the enzyme, we get a glucose. So glucose now can diffuse out by facilitated diffusion and then increase the blood glucose level. Are you clear? So get this. So what's mean cell signaling? Cell signaling actually involves here. Okay, but inside the liver cells, you do have this process. It's known as signal transduction. So signal transduction require ATP. In this process, ATP is required. I will show you guys why later. Okay, for signal transduction, ATP is needed. Okay, but in our topic, cell signaling actually includes the communication as well as a signal transduction. Are you clear? So a bit confused here. Don't worry about it. Signal transduction, okay, cell signaling in our process generally includes the entire process. We term it as cell signaling. But the one that requires ATP is a signal transductions here. Are you clear? Where the glucagon acts as the primary messenger. Okay, so it means that later we're going to generate secondary messenger. Okay, so until this part, clear or not? So we detect low blood glucose level. We want to increase back the blood glucose level. Definitely, we need more glucose. So in this case, we need to have the cell signaling and then inform the liver. How to inform? We use the chemical signaling molecule. In this case, the glucagons, the water-soluble ligand. Okay, and we want specific. We don't want the whole body response. So it means that we have specific receptor. So this glucagon receptor only appear on the liver cells. But the thing here is my signaling molecule, glucagon, is a water-soluble hormone. Can they directly enter into the cells and then tell the enzyme, hello, enzyme, activate now? You cannot. Are you clear? So they have to bind to the glucagon receptor, trigger a series of reactions until we get this enzyme to be activated. And then with the enzyme activated, we will be able to get the hydrolysis of the glycogen. So in terms of this process, we have three very important steps here. So first, step number one, okay, binding of ligands, means hormone, to its specific binding site. On the receptor. Number two, step number two. This step step involved. Step number one, generally, Step number two. So the binding will cause a series of reactions until the productions of second messenger. Okay, and because of this second messenger, you're going to involve the third step, cascade activation of downstream enzyme or protein. These are the three general steps involved in the signal transduction. So first thing, the ligand have to bind to the specific binding site on the receptor. So this receptor depends on the ligand, because if the water-soluble ligand, this receptor will be on the cell surface membrane. Okay, no point you put the receptor inside the cell, because you put the receptor inside the cell, useless. Why? Because our ligands won't be able to enter. But if the ligand is a lipid-soluble ligand, and then no problem, receptor can be inside the cell. Done for this one? Okay, Okay, let us look at this membrane receptor. Okay, when we talk about membrane receptor, we also term them as the receptor protein that bind to the signaling molecule and initiate the physiological response. So generally, they are transmembrane protein. So they have the ability to bind to the signaling molecule outside of the cell, subsequently transmit the signal through a sequence of the molecular switches, so on and off. And they must be very specific. For example, dopamine receptor only can allow dopamines to bind. Insulin receptor only allow insulin to bind. Are you clear? So membrane receptor interact both extracellular signal and molecule within the cells. Okay, so allow this signal transduction to take place. And it's very important because most of the signaling molecule, they are too large or polar or charged, okay, to cross the cell plasma membrane. So therefore, we need to have this membrane receptor. Okay, and this membrane receptor also provide what we call the specificity. Okay, because it depends on the type of the cells. Not all cells will express or have that particular proteins. Are you clear? Okay, so once the receptor protein receive a signal, this is very important process. When it receives the signal, it will undergo conformational change. Conformational means that the shape, right, the precise three-dimensional shape will change, and it will launch a series of chemical reactions. I see that, and this process involves the production of second messenger, and we call them as a relay signal. Now, what does this mean? So it means that what is our first signal? Our primaries. Our primary messenger will be our ligand, but ligand cannot enter. Can you see that? Ligand cannot enter. So try to imagine it's something like you online. Okay, online. I mean, what you call this? Shopping. Okay, online shopping. So when you're online shopping already, so the delivery guys send something to you. So where they put in, I mean, let's say for example, like letter, for example, you put in your post box. Can you see that? So the post box is a receptor. Can you see that? The post box is the receptor. So what happened next? Someone going to take this to you. For example, you are in a room, for example. Okay, your mom going to take it. So your mom will be the second messenger, pass this item to you. Can you see that? Because the postmen or the delivery guys won't straight away enter into your house and pass the items or the products to you. So someone have to help you to carry into your room. So those are second messenger. So we have three major classes of the second messenger. We have the cyclic nucleotide, which is CAMP. This one you have to know. Highlight. Okay, and the two not in our syllabus. These two are not in our syllabus. You do not need to know, but I will briefly tell you guys about this inositol triphosphate or IP3 and DAG pathway that involve the PIP2. Okay, so this one not in our syllabus, just for your informations only. So if you want to know more, please feel free to go through it. Okay, so membrane receptor also, we can divide them into few classes, and our topics will focus in this G protein coupled receptor, or we term it as GPCR. Okay, G protein coupled receptor. So it means that this receptor actually linked together with G protein. So the activation of G protein allow a series of reactions to take place. Here. So let us look at this G protein coupled receptor. So just now we said that the receptor, they are transmembrane molecule that couple together with the G protein. So that's why we call it G protein receptor. Is the most common, a big group of the cell surface receptor, the largest family. Okay, a lot of the ligand actually utilize this G protein coupled receptor. In our syllabus, we have three of them. And in A2, we're going to learn first glucagon receptor. Number 2, adrenaline receptor. Number 3, ADH receptor. They are all fall into this GPCR. Okay, so there are hundreds of different kinds of GPCR, and nearly a third of all drug target these types of receptor. Are you clear? So a diverse cell ligand bind to this receptor, including water-soluble hormones and neurotransmitters. So our today focus will be water-soluble hormones. Are you clear? So coupling with G protein, they are called 7 transmembrane receptor because they pass through the cell membrane 7 times. So 1, 2, 3, 4, 5, 6, 7 times. So they have extracellular N-terminus. So N-terminus means that the starting point, and intracellular C-terminus. Are you clear? So this C-terminus will join to the G protein. Can you see that? G protein? G protein is the heterotrimeric protein. Now, what's hetero? Hetero means different. Okay, trimeric means 3 units. Can you see that? We have the G alpha, we have the beta and gamma. So the entire protein is a quaternary protein. Are you clear? They are quaternary protein. So this quaternary protein with 3 subunits. And the most important part here is the G alpha. G alpha has the binding site for GDP. Are you clear? Can you see that the GDP? Okay, so G protein composed of 3 different subunits, which is alpha, is the one that we need to learn. G alpha proteins, beta and gamma. Okay, 3 of them. So alpha subunit bind either to GTP or GDP. Can I see that GDP? GDP means that when this receptor is inactive. When they're inactive, you can see that they bind to GDP. But when it's active, GDP will be substituted by GTP, then it becomes active. So means that something already bind to this receptor already. So our ligand, okay, or agonist bind to. So don't need to worry about agonist at this moment, guys. Too many terms for you guys. Okay, so ligand. When ligand bind to the receptor, it's going to change the shape of the receptor and activates my G protein. How to activate? We change the GDP to become GTP. So GDP is something like our ATP. Are you clear? Something like ATP. So it means that with the GTP, you can see that now my G protein dissociated, this one become active. So when G alpha become active, it's going to cause a series of reactions. Are you clear? So 2.16 show the interconversion of active and inactive G protein. So this inactive, this is active. Active, you can see that with GTP. So now we have this G alpha now become active. Okay, so GPCR, what is the functions? Basically, they transduce the extracellular stimuli to give the intracellular signaling. So they involve in what we call the signal transduction. Okay, through the interaction of their intracellular domain means binding site with the heterotrimeric G protein. So what I mean intracellular domain? Binding site, intracellular domain, intra means within the cell with the G protein. So therefore, they will be able to activate the G protein. There are 2 principal signaling transduction pathway involved in GPCR. In our syllabus, you have to know CAMP signaling pathway. You cannot run away. This is in our syllabus. GPCR will be CAMP signaling pathway. Another one is the PIP2, what we know as a Phosphotidal Inositol signaling pathway. This one I will show you, but you just need to know it's for your information only. As I said, that question can come out with anything, but if out of syllabus, they will give you sufficient information. Okay, so let us look at CAMP pathway, which is in our syllabus. Now look at this diagram. Complicated, right? Okay, but don't worry. As I say that at this moment, no need to worry much. Just need to know in just what we call this the brief one first. Okay, so first thing, we have this GPCR. Okay, so this GPCR is the epinephrine. Epinephrine, another name is called adrenaline. Okay, adrenaline is the fight and flight hormones. What means fight and flight hormone? Means that you are in crisis. When you are in crisis, you want to run away or you want to fight back. The most important thing is you must have sufficient ATP to run away. You must have sufficient ATP to fight back. So in this case, we need to have a huge supplies of glucose. Correct or not? So it means that where is our glucose storage? Our liver cells. Liver cells have the glucose storage in the form of glycogen. Can you see that in the form of glycogen? So therefore, in this case, we need to release the glucose from where? From glycogen. Okay. So how this GPCR actually functions? So first process, as I say that, adrenalines or epinephrines have to bind to the specific receptor. Okay, the binding site of the receptor on this receptor protein or epinephrine. And then the binding of the ligand, the epinephrines or adrenalines onto the binding site, going to activate the G protein. Why it can activate? Because the binding of epinephrines or adrenalines onto this receptor will change the shape of the receptor. So the change of the shape of the receptor actually allowed the activations of the G protein. Can you see that? I say that inactive, you can write down, inactive GDP. So now become active. So activates the G proteins. So become GTP. So once it become activated already, so these G proteins now continue to activate who? They activate one of the enzyme called adenylyl cyclase, this enzyme. Can you see that? This enzyme actually located where? On the the enzyme is anchored at the cell surface membrane. So what this adenylyl cyclase do? Adenylyl cyclase actually converts the ATP into the CAMP. So CAMP is actually a second messenger. Are you clear? It's a secondary messenger or second messenger. So epinephrines is our primary, the first messenger. Second messenger is a CAMP. What CAMP can do? Now look at this, the number here. The number tell you something. We learn in enzyme. So that's why we need to learn enzyme first. We learn in enzyme, in order to increase the rate of reaction, we always increase the number of enzyme available. So it means the enzyme must be in high concentration, correct or not? So that my rate increase. So in terms of this fight and flight hormones, you need to actually make sure that you have a lot of glucose. So in order to have a lot of glucose, my enzyme must be in high concentration. So how we do this? So we need to actually having the cascade activation. So what is the cascade activation? Cascade activation means that, look at this, look at one molecule. So this is only one receptor bind to one adrenaline. From one adrenaline, because of the enzyme have high turnover. We learned about this, right? Enzyme has high turnover. So it means even we only need to activate one G protein. This one G protein activate one adenylyl cyclase. One adenylyl cyclase per unit time can give us 20 molecules of secondary messenger. Can you see that? Are you clear? So because of the turnover, definitely you cannot compete with the carbonic anhydrase. You definitely cannot compete with this carbonic anhydrase. We have 600,000, 600,000. This one, not so many, okay? Not so many, 20, but enough already. Why? Because you can see that cascade activation means that we amplify the signal now. We amplify the signal from one only, one. Can you see that? I got 20 CAMP. So 20 CAMP going to activate the protein kinase A. PKA, protein kinase A. 20 of them. So protein kinase A is enzyme, right? It's an enzyme. So this enzyme continue to activate who? Activates the phosphorylase kinase. For every 20 of them, they activate hundreds of them now. Can you see that? Means that I get more and more. Can you see that how many fold? From one fold, we have 20 fold of the CAMP. From 24 CAMP, activate 20 of this protein kinase A. From protein kinase A, we're going to continue to activate 5-fold each of them. So become 100. 10-fold. Can you see that? Continue. So means that with one epinephrine or adrenaline, I managed to activate how many? 10,000 of the final enzyme, which is glycogen phosphorylase. Can you see that? Glycogen phosphorylase is the enzyme that breaks down glycogen into glucose 1-phosphate, and which will be dephosphorylated to get glucose, and glucose will be released into the blood to increase the blood glucose level. Are you clear? Okay. So means that the most important enzyme here is the protein kinase A. Once the secondary messenger CAMP activates the protein kinase A, we are going to get a series of the cascade activation. Cascade means that we amplify the signal. So at the end, with 1 unit of the adrenalines, we can get 10,000 units of the final enzyme, which give us the glucose that we need for the crisis that we could either fight or flight, okay, for adrenalines. So now take a bit of time, 5 minutes to complete this passage using the given term now. Okay, so since you guys have done, so let us look at the answer here. So the binding of what? The binding of the ligands, okay, such as adrenalines to G protein coupled receptor activates the attach G protein by stimulating the exchange of GTP for GDP. Cannot. We add GTP, we remove GDP. Okay. So the activated G protein then dissociates into the G alpha and a beta gamma complex. Okay. So means that beta gamma dissociates from the G protein G alpha. So G alpha now is activated. So G alpha bound to GTP is active and can diffuse along the membrane surface to trigger the synthesis of the small molecules known as second messenger, okay? So which initiates and coordinates intracellular signaling pathway. For example, cyclic AMP is a common secondary messenger or second messenger involved in the signal transduction cascade. So the CMP synthesized for ATP by the enzyme adrenalyl cyclase, which resides in the cell membranes, okay, located at the cell membrane. So the activation of adrenalyl cyclase can result in the synthesis of hundreds or even thousands of CAMP molecules, because not per unit time, now overall. Okay, so this CAMP molecule activates the enzyme. CMP activates who? Activates the protein kinase A. This is very important, enzyme protein kinase A, which then phosphorylates. Okay, kinase. Always remember, kinase is the enzyme that helps to phosphorylate. Okay, we have another one called esterase. Esterase is the one that removes the phosphate groups. Okay, so for us, we just need to know the kinase. Kinases actually always involved in the phosphorylations. Okay, multiple protein substrates by attaching phosphate group to them. Can you see that? So each step in the cascade is further amplified. Further amplified the. Oh, sorry, what happened? Okay, sorry. So cascade further, further amplified the initial signal, and the phosphorylation reactions mediates both short and long-term response in the cells. At the end of the process, the CMP molecule are hydrolyzed by the enzyme. Okay, esterase. Can you see that? Esterase is the one that breaks down to stop the signal transduction cascade. Okay. So what is the first step? The first step here. Okay, the first step involved here, basically, we do have the ligand bind to the receptor. Okay, so first step. So in general, I say ligands, this water-soluble ligand binds to the specific binding site. Okay, bind to the specific binding site on the receptor. Okay, so what is next? So G protein now is activated. So G protein activated, G protein now activate the adrenal cyclase. So become activated. Okay, so the binding here. Okay, so something extra that we need to know. The binding of the ligands to the specific binding site on the receptor. First, it will change the precise three-dimensional shape of the receptor. And this change of the three-dimensional shapes will cause us to activate G protein. Particularly in this case, it's G alpha. Are you clear? But you don't need to be so precise. Okay. So step number 2. So what is the step number 2 in this case? Step number 2. So G protein now, the activated G protein activates the enzyme. So this enzyme is adenylyl cyclase. Okay, so this is the second process. So what's the third process? The third process involved here. So it's enzyme. So enzyme do what? Enzyme actually produce a second messenger here. So from ATP becomes cyclic AMP or CAMP. So CAMP act as second messenger. Okay, so this is the third process. So write down here the third process. So what's the third process? So this adenylyl cyclase activated okay, so the activated adenylyl cyclase now catalyze or converts ATP to CAMP. So this CAMP is the second messenger. Are you clear? Okay, so what this second messenger will do? Second messenger actually now we're going to involve. Okay, second messenger will involve in the cascade activations of downstream. Downstream means that after that one, okay, downstream either protein or enzyme. So what is the enzyme that involved here? So this cyclic AMP now involved in the activations of protein kinase A. Okay, so protein kinase A, pKa. So this is inactive. Now become protein kinase A, which is active. Okay, so this pKa active then continue to activate another enzyme, until we get the final, okay, and enzyme to be activated. So this final enzyme, when it become activated already, then we can see that we get the effect. So this effect also termed as cellular response. Okay, cellular responses. Clear? Are you clear? So what is the functions here? So now number 4 here. Step number 4. So CAMP now activates the protein kinase A. Okay, protein kinase A. So result in signal amplification. So what is the effect of signal amplifications? The effect here for signal amplifications, we get high concentrations of enzyme. So therefore increase the rates of reactions and allow us to have cellular responses. Okay. So so this diagram summarizes the entire process now. Okay. So I say that this is the what we say that the overall process. Can you see that the signaling molecule activate relay molecule? So we have the CAMP, for example, and then we have the different active protein kinase, and then after that we get the cellular response. So this we call phosphorylation cascades. Okay, in general summarize. Okay, the phosphorylation cascade is a type of signal transduction. Now another pathway is termed as Phosphotidal Inositol signaling pathway. Okay, so this is a little bit more complicated. I just go through it briefly with you guys, okay, because you are not required to know, but the question can give you this diagram and guide you step by step. Number first one, who is the primary messenger, who is the secondary messenger, and so on. Are you clear? Okay, so now look at this. In this process, again, we must have the receptor, also G protein. Can you see that? The G protein. So the alpha bind to the GDP. Can you see that? Alpha bind to the GDP. So these no epinephrines or no adrenalines bind to the receptor. So activate the G protein as usual. Okay, so G protein actually now, instead of adrenalyl cyclase, what will happen here is, can you see that? We have phospholipase C. So this phospholipase C going to cleave of this PIP2, degrade. Can you see that? Degrade PIP2, make it into two different compounds. The first compound is called DAG, another compound is IP3. Can you see that? Are you clear? So this GTP means that my G alpha become activated. So previously, G alpha or G protein, the activated G protein actually activate the adrenalyl cyclase, kind of in CAMP. But in different cells, they activate different things. So they activate phospholipase C. So with the phospholipase C, what will happen here? Phospholipase C is an enzyme to cleave, means that, okay, break down the PIP2 into the DAG and IP3. So DAG, we're going to activate the protein kinase C and give us the downstream effect. And IP3 will cause us to have the calcium ion. So calcium ion can activate other enzymes. So this one actually involves even more responses. Can you see that? Compare the CAMP, we leads to one response. But this one can lead to many, many responses. Can you see that? Okay, because we have two secondary messenger now. What is the two secondary messenger? DAG and IP3. So each of them have one function. IP3, water soluble. DAG, they remain in the cytoplasm, activate this protein kinase C. So when activate protein kinase C, then you can see that you will have the activation from the normal protein become phosphoprotein, means that they get the something like kinase. Kinase basically means they involve in the phosphorylation. Transfer the phosphate group to the protein. Can you see that? The protein become phosphoprotein. Okay, and also in this case, you can see the endoplasmic reticulum here release the calcium ion. Calcium ion will activate other kinds of the enzyme. Okay, so this one I will go through. Okay, you can try it. Okay, 5 minutes. Okay, let's continue to see what are the answer. What's the correct answer? So beside the cyclic AMP signaling pathway, another important signaling transduction pathway use a GPCR. Okay, is the Phosphotidal Inositol signaling pathway, or sometimes called as Phosphotidal Inositol cascade. Okay, again, it's a cascade. For example, the angiotensin 2 receptor of our body use this pathway to regulate our blood pressure in the cardiovascular system. Okay, because blood pressure is something to do with the muscle contraction. Calcium ion also involved in the muscle contractions. Okay, so phosphatidylcholine signaling begins with the binding against the primary messenger or the ligands, okay, to the specific binding sites on the GPCR. Okay, so the bindings induce conformational changes, still the same. Can you see that? Okay, that results in the G protein to expel the GDP, remove GDP, bind with GTP, still the same. So this causes the G protein to detach from the rest of the receptor, okay, move on to and bind to the membrane-bound enzyme, in this case, no longer adrenalyl cyclase, but phospholipase C. So upon the binding of phospholipase C, the G protein stimulates to begin to break down the PIP2. Can I see that? Right? PIP2 into two secondary messenger molecule, which is DAG and IP3. So the one that remain within the membrane is DAG, okay? It's a non-polar and remain within the membrane of the cells. It moves along the membrane and bind to another membrane-bound enzymes known as protein kinase C. Okay, so this IP3, okay, on the other hand, IP3 is a water soluble. That's why they can move into the cytoplasm, bind to the ligand-gated calcium channel on the ER membrane. Okay, so it means that this what does mean ligand-gated calcium ion channel? So we learn about this ion channel. They can have the gate open and close, correct or not? So it means that with the IP3 can cause the opening of the gated calcium ion channel. So it causes the calcium ions, okay, to rush into the cytoplasm. Here, so the calcium ions have two functions. First, it can bind to the protein kinase C, and with the help of DAG, stimulate and activates the kinase. Second, the calcium ion can bind to the comedonins, forming the this complex that goes to activate effector protein and enzymes such as protein kinase. So this kinase can then phosphorylate other protein enzyme channel and pump that ultimately carry out the cellular responses. So this one no need to know in detail, just the way for your information only. Okay, so now next part. So we have done for the water soluble. We have done for the water soluble ligand. How about lipid soluble ligand? So for lipid soluble ligands, then you can see that because lipid soluble means that they can diffuse across the phospholipid bilayer crema. So means that intracellular receptor are receptor protein found inside the cell. Okay, so can you see that? Intracellular, intra means within. So intracellular receptor are receptor protein found inside of the cells, typically either at the cytoplasm or nucleus. Okay, so it means that in this case, for this kind of intracellular receptor, the ligands are small hydrophobic molecules. So it means that they can diffuse across the plasma membrane in order to reach their receptor. Okay, particularly you can see that for the sex hormones, which are steroid hormones, or we call it lipid soluble hormone. So for these lipid soluble hormones, okay, they are intracellular. So what is their signaling pathway? So in this case, look at the figure 2.20 here. So we have the plasma membrane, we have nucleus. So in this case, for most of these lipid soluble ligand cell signaling pathway, they must form this receptor hormones complex that manage to enter into the nucleus, or already this receptor inside the nucleus. Now look at this. Signaling molecules, because they are lipid soluble, then they can enter into the cells by diffusion. So when enter into the cytoplasm, you can see that they have the intracellular receptor now bind to the signaling molecule. So this signaling molecule is a hormone, then it becomes hormone, receptor hormone complex. So this receptor hormones complex now can enter into the nucleus, bind to our DNA, and cause the transcription to take place. Like what I explained it just now. Okay, for the pKa's, right? pKa bind to other protein to form a complex. But in this case, you do not need to go through the pKa. The hormones or lipid-soluble signaling molecule directly bind to the complex and bind to the receptor to form a complex, which can result in the transcriptions, allow us to have the mRNA. So this mRNA will be translated to form polypeptides. Are you clear? Okay. So what are the differences? What are the differences? Now look at this. Okay, so first, differences in terms of where is the receptor. the main. So for water-soluble ligand, it must be on the surface membrane or cell surface membrane. It cannot be inside. Okay, but for lipid-soluble ligand, now free. So it means that they can add the cell surface membrane, no problem. It can be inside the cytoplasm, no problem. It can be inside the nucleus, no problem. Are you clear? This is the main locations of the receptor. Okay, so this is the main one difference. Then definitely we have many. Okay, we have many. For example, secondary messenger. Then for water soluble, then yes, you have production of second messengers, but lipid soluble ligands, no secondary messenger. Useless. Why? Because my signaling molecule, can I see that my signaling molecule can directly diffuse through the, can I see that? Diffuse through this surface membranes. Can I see that? Diffuse and then bind to a receptor. This receptor can be inside nucleus. Can I see that? Gene regulations and give a cytoplasmic response. And just now you can see that in terms of this, you can have the cellular response straight away, or you can form a complex, have a gene regulations. So this part, the first arrow here, can I see that the highlight one? To activate the enzyme. This one to activate the gene. Activate enzyme, activate genes. Okay, so these are the two main differences. Okay, we have more, but I'm not going to talk about it now. Okay, these two enough already for you guys. Okay, so with this, I have done for today topics. Okay.